Yes, sir. So yesterday's class, I was about to tell you, or rather, I was explaining you that how a protein can be made. That is, the central dogma says that the DNA is the base of making protein. The codons are found, or rather, the information about what type of protein has to be made is found only on the DNA. But the question was, when this group people understood first only the central dogma, that is the DNA made protein, but there was something missing, because DNA is double stranded, and protein is the polypeptide. Protein, that is the polypeptide, is single chain. Moreover, in DNA, there are only 4 types of nitrogen bases. 4 nitrogen bases are found, 4 kinds of nitrogen bases are found. They are adenine, thymine, cytosine, and guanine. Whereas there are 20 different types of amino acids are there. Okay? There are of 20 types. Now, 20 types of amino acids has to be coded by 4 nitrogen bases. So this was a very big problem. How the double-stranded DNA will code a single stretch of the single strand or single chain of the polypeptide. That was a big problem. And how 4 nitrogen bases will code for 20 type of chemistry. So on searching, they came across another molecule called RNA. And RNA is a much smaller molecule which has got single strand and the same kind of 4 nitrogen base. The only difference is that in case of thymine there is uracil. So adenine, uracil, cytosine, guanine, cytosine, uracil, adenine. Whatever can be the combination, differ from one gene to another or one polypeptide to another. So this is how DNA first make RNA and RNA make protein. This thing is coming to become clear. That is, the double-stranded DNA will make another type of polynucleotide chain which has a single strand, that is the one single strand, and the single strand can easily copy a single chain of polypeptide. So one problem was solved, that is how double-stranded DNA can make a single strand or single chain of polypeptide. Now the second problem was, that is how 4 nitrogen bases, if I write 4 nitrogen bases here, so the 4 nitrogen bases are adenine, uracil, cytosine and guanine. Adenine, uracil, cytosine, guanine. How 4 nitrogen bases will code for 20 amino acids? This was a very big question. And to understand this question, the scientists from various disciplines have been used. There was a scientist, George Gamow. He was a physicist. He argued that a 4 nitrogen base can no way code for 20 amino acids if they are taken in singles. So, according to law of collision, he said 4 to the power 1, that is only 4 combinations are there. 4 to the power 2, that is 16 combinations will be there. 16 combination can also code only or control only 16 amino acids. 4 will still remain uncontrolled. So he said the codon should be found in the form of 3. That is, there will be a possibility of 64 codons. So if the nitrogen bases are taken in a set of 3, they will be known as a codon, and there will be altogether 64 codons possible. This was only a theoretical observation which he made. He said it should be like this, but then it should be proved. Unless it is proved, how can it become a law, or how can it become a truth? So the people from the different scientists, different fields were again engaged, and we come across here some of the names. If you read page number 111, last paragraph, providing the proof that the codon is a triplet was more daunting task. The chemical method developed by Hargobind Khurana, an Indian scientist who was working in US, instrumental in synthesis of RNA molecule with definite combination of bases. What he did, he could artificially make the RNA with definite combinations like A, C, G, whatever he wanted, those type of amino acid he could bring together and he can make. And then he studied that what type of proteins are made by this, but that time the protein synthesis was not artificially possible. So then again there were different scientists came in play. Next was Marshall Nierenberg. He was the first to produce protein outside the cell. Marshall Nierenberg was a scientist who could produce cell-free synthesis of protein outside the cell, synthesis of protein. That was very important. So Hargobind Khorana produced the RNA chain with different type of combination, and those chains were used to make protein by using the method of Marshall Nierenberg. And next is Cerevo Oka. He actually produced some enzymes which were required for this type of protein synthesis. So the scientists from all different disciplines, that is physics, Hargobind, John Gamow was used, that is his ideas were used, then Hargobind Khorana, then Marshall Nierenberg, then Cerevo Oka. All these have put their contribution, and many more must have put the contribution. We are not aware because every name cannot be mentioned in the book. And it's a slow process because it's going in the early 19th century, which was a very, that time the knowledge about the biochemistry was very much limited. So we never had so much of opportunity to use them. Okay, so finally it came into being that a codon, genetic code, should be consist of 3 nitrogen-based sequences. So now what things are clear to you? Yeah, what things are clear to us? I am mentioning here. That is genetic code. What is the genetic code? A code which decides the sequence of amino acids in a polypeptide chain. So now picture is a little bit clear. We have got DNA, very large molecule. From DNA, a certain segment to certain segment is copied in the form of RNA. That is called as a gene. That copying of the DNA to RNA is done, and then RNA has a sequence of various types of nitrogen bases, and these nitrogen bases sequence will act as code which will decide that what type of polypeptides are formed. And we already earlier studied that there are 3 different types of ribosomes, that is the RNA that is used, that is tRNA, mRNA and rRNA, which will be used in the protein synthesis. So the codons, that is 64 codons, how can we write the 64 codons? It is, of course, it is not in codes, but I am just explaining you the method how it is being written there. We have got 4 columns. And we write A, U, C, G, A, U, C, G. So here A from here. And the third one is A from here. Then U, third is A from here, C, then fourth is A, G. Then the same way U, U, then third is U, C, then U, G. Like this the codons are made. And by the study of these codons, we came to know that what each codon stands for. And before that, I'll explain you some of the salient features of the genetic code. So it is mentioned here on the page number 112, the salient features of the genetic code. First, the first point of our first salient feature is the genetic codes are used in the form of triplets. 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. That is, genetic codes will always be in the form of triplets. But not all 64 codons will be used for defining amino acids. Only 61 codons are functional, which defines amino acid. 3 codons do not have any respective amino acid. So they are also known as nonsense codon. They do not have any meaning, they do not have any sense. So that is why they are also known as nonsense codon, or these codon, whenever they are present in a polypeptide chain, the process will terminate there. The protein synthesis will terminate there. So they are also known as stop codon. They are also known as stop codon. The stop codons are only 3. They are UAA, UGA or UAG. These 3 codons do not have any respective meaning. So if in a RNA this is there, the tRNA will not be able to read them and the process will be terminated there. Okay, so that is why they are also known as stop codon or nonsense codon. Okay, so the first point or first salient feature about the codon is that the codons are found in the triplets, and if we take the 4 nitrogen bases in the form of triplet, there will be 64 combinations, 64 possibility. Out of the 61 codes for codon, amino acid, and 3 remains unexpressed, or they do not have any meaning, or whenever they are there in the messenger RNA, the process of protein synthesis terminates there. So they are also known as nonsense or stop codon. And the stop codons are UAA, UGA or UAG. Only these 3. The next property is written, that is the next salient feature written, is that the codon codes only for one codon code for only one amino acid. One codon codes only for one amino acid. If I read from this table over here, the triple U, that is UUU, no matter where you are taking, it will always code for phenylalanine. Triple U will always code phenylalanine. It will not code for anything else. CUC is given there, that is to stand for leucine. So leucine is a type of amino acid which will be coded by that. Okay, so one codon will code only one type of amino acid. So that is a very important property, that is called specific. This is a specificity, so it is specific nature of the codon, or it is also known as unambiguous nature or property. The third one is some amino acids are coded by more than one codon. Some amino acids have more than one. If you look at the table of the codon over here, you can see that the leucine is coded by 6 different type of codons. Leucine, the first column right on the top, you can see the third number, it starts UUA, leucine, then again UUG, again stand for leucine, then CUU, leucine, then CUC, leucine, CUA is leucine, and the fourth one is CUG is also leucine. So one amino acid can be coded by more than one codon. There are several amino acids which have 2 codons. There are some which 4 codons are there, and for leucine like 6 codons are also there. And if you look at their arrangement, their structure, you will find that the first 2 codons are in all these cases are constant. The last one is bringing the difference. That is if you have to have phenylalanine and you have to have leucine, the difference coming only in the third one, that is UUC will code for phenylalanine and UUA code for leucine. So difference is lying on the third one. Okay, and in case of leucine, we will say that is UUA, UUA, UUG, CUU, CUC, CUA and CUG, all of these are standing for leucine. So one amino acid can have more than one codon. Okay, so that is called degenerate property. This property is called degenerate. Now the question will come in the exam like this way. What do you understand by the degenerate nature of the codon? So you have to write that one amino acid can be coded by more than one codon. This property of the codon in which one amino acid is coded by more than one codon is known as degenerated nature. And when it comes to specific nature or unambiguous nature, then you have to write in the just opposite to it. That is, one codon will always code one type of amino acid. Okay. Then the fourth property says that when they are read in when the protein synthesis takes place, these codons will be read in a contingent form. That is, there will be no amino acid, no nitrogen base will be skipped. Suppose this is the messenger RNA, and here it is UA, AUG, CCC, GGG, UCG, or I like simply UUU, then again AAA. Some you have to learn. Okay, and suppose here it is UAA. So this is one codon, this is another codon, this is third codon, this is fourth codon, this is fifth codon, and this is nonsense codon. You cannot have this way, that is this AUG we have taken, then this C we skip, and we get the combination of this C and G, and then GGU, and again we skip this one and UAA, not like this. You cannot skip any nitrogen base in a codon. They will be always read in a contingent way. Like in some of the book it is mentioned as there is no punctuation present or found in this. As when we read the sentence, we follow the punctuation, like comma is there, full stop is there, colon is there, semicolon is there, inverted commas are there. So accordingly we make our sentence read. But here there is one law, that is they should go continuously, one after another. No skipping of any nitrogen base in the polypeptide as well in the messenger RNA. So that is again a very important property. Next is, a codon is universal, nearly universal. What does it mean? Right from the smallest organism, that is from a bacteria, to the most complex organism, human being, most of the codon are exactly same. This is one of the important evidence that we have evolved from one another. We have evolved from one another. That is, bacteria comes in prokaryotes. From the prokaryote to the most highly developed eukaryote human being, the amino acid or the codon AUG will always stand for methionine. There is no error anywhere. How? Now nearly it is written, it is not written always. However, there is some variation found in protista. There are some variation found in the protista and chloroplast and mitochondria. The mitochondrial codons and the chloroplastal codons and some of the codons found in the protista show some different behavior. Otherwise, right from the prokaryote, bacteria to eukaryotic, most developed organ, human being, codons are always universal. The differences except this. These three places the variations are there, differences are there. I can read there. What is written that the codon is nearly universal For example, from the bacteria to human being, U code for phelanine. It is always coding for the same thing. There is no variation, no difference. Okay, so that is very important property. That is very important property. And that gave the evidence that we have evolved from one another. Had we evolved in a different manner, like what you say, you can put it this way, that is the world over, different societies are there, different cultures are there, different ethical groups are there. So the way we consume wheat in Rajasthan, in older culture, Rajasthanis, they used to simply knead the atta, they used to make the bati, they used to bake it in the ash and they used to eat. But now it become near universal in the all over India, that is they all eat the atta, they make the chapati, they bake it and they eat it. So we are eating wheat in the form of chapatis, but it is not in the Western culture. They are making bread out of it. If you go to the Middle East, you will find that other type of chapatis, Doritos and something else. So that depends. Now that is saying that we have not learned the making chapati from the same origin. Had we learned it from the same origin, it would have been same all over. But in due course of time, that is changed. But we have the same origin, that is the human being evolved from the same Adam and Eve. So from that time to this now our time, we got only 2 eyes and their locations are there, 2 ears, 1 nose, everything is same. So if we have same origin, our things will be same. If we are different origin, like chapati making or consumption of wheat, are the different origin, different culture. Some started making cake out of it, some started making pasta out of it, some started making porridge out of it, some started making halwa out of it. So they have got different culture. Because in due course of time these things are done, they are learned in a different manner. But those things which are the same common origin will always have same. So here it is showing, it is forcing that we have to believe that from the bacteria till human being, there is not a single difference except in some of the proteins. They must have got some mutation in due course of time. And chloroplast of the plants and mitochondria of biology. You know that chloroplast and mitochondria have got their own DNA, they are not depending on the nuclear DNA. Is it clear to you? Have you ever heard of it? You must have studied in the class 9. That is the DNA and chloroplast and mitochondria have got their own DNA. They are not depending on the nuclear DNA for their function. So their DNA have different type of codes. Okay. And the 6th property which is given there is the AUG is dual nature. If it comes in the beginning, it is starter. And if it comes anywhere in between, here suppose it is coming AUG, then it will be coding for methionine. AUG has double nature. When it comes in the beginning, there is a very long chain. And in this, there are different codons. 1, 2, 3, 4, 5, 6, 7, 10, 20, 25, 60, 70, 100, 200 codons may remain useless. And then one codon, that is AUG, comes. So the protein synthesis will begin from this point only. These all will be left unused. Okay? And from here the process will begin. So AUG is search, and from there the process begins. So AUG is also known as starter. And whenever it will come in between, anywhere, then it will be standing for methionine. So these are the 6 important properties. The question will be asked in the same manner. It will be asked, what is the starter codon? What do you understand by the universal nature? What you will be understanding? What do you understand by degenerate nature or unambiguous nature or specific nature, etc. Is it clear? So the codons are total in 64, 60, there should be 64 codons, but after that only 61 are coding for amino acid and 3 are coding for stop codons. They are called stop codons or they are called nonsense codons. They are not standing for any amino acid. And then we have studied the 6 properties This was actually the first property. Then the second property, that is each codon is standing for only one amino acid, right from the bacteria to human being. There is no change. Change lies only in these two categories. So this is on the unambiguous or specific nature. Then we have studied that one amino acid may be coded by different, like if I can put it this way, like a child may have different names. His mother may be calling him by different name, or father may be calling him by different name. The school teachers are calling him by the original name which is there, or sometimes they simply call him by only the surname, or the friends call him by different name. The same amino acid can be coded by different name, different codons. Okay, but whatever is the one codon is there, that is only for him. Okay, like in a class, if we have Aishwarya Sharma, then Aishwarya Sharma is only one person. Okay, if two are there, then we use their father's name. If they are also same, then we use put 1 and 2, whatever. But we have to differentiate. So one name is given only for one child. Similarly, one codon for only one amino acid. But the third property is just opposite to it. It says that one amino acid may have many codons, right? One child may have many names. Then nearly universal nature and then name. So this is what we have studied. Now we come to the interesting point. Codon we have come to know, the behavior of codon we have come to know. Now we are seeing something. Suppose I got this as a amino acid. Sorry, RNA, and this RNA molecule, A will starter, U, A, C, G, A. So what type of polypeptide it will code for? If we look at in the table, we can come to know. If we look at the table, we will come to know. It is actually started, just leave it. This is standing for phelanine, PHE. Triple A is standing for, triple A is standing for lysine. Triple C is standing for C and C. Okay, CCC is standing for chlorine. That is a brand. Chlorine shirts and brands and all. And triple G, arginine, and triple A again lysine. So looking at the messenger RNA codons, we can define that what type of polypeptide chain we will get out of it. But suppose you are given a sequence of amino acid. If we want to make this amino acid with these combinations, arginine, and we want to make a polypeptide of this combination of elements. Now you define a messenger RNA. If this thing is given to the 10 different children, all 10 will give different combinations of the codon. Because the lysine can have more than one codon, the chlorine can have more than one codon, arginine can have more than one codon. One child, whatever codon is writing, will not be necessarily be used by the same by the other child also. So this is why it is easy to understand, or it is easy to define the kind of amino acid from RNA sequence, but we cannot get the other way, that is from the polypeptide chain. If someone asks you to define an RNA chain for it, it will be difficult for us. Okay, so this is what enough for today. Tomorrow's class will study about mutation. Okay, thank you children.